circuitry. Excellent. And they can remodel this circuitry and ensure that it forms properly so that the CNS develops as it should. They can also secrete a number of different cytokines and chemokines. You can see down here one example where they are secreting OPC survival factors so that these oligodendrocyte progenitor cells can then differentiate into myelinating oligodendrocytes, which leads to myelination within the developing CNS. As development finishes up, though, microglia kind of switch up their roles. They turn into this, well, they don't turn to anything. They're still microglia, but their roles kind of change. So again, in adulthood, we see this role of phagocytosing myelin and pruning myelin to make sure that it's at its optimal ensheathment, can phagocytose other cells. And one characteristic of microglia that's really neat is that they're constantly monitoring neuronal activity and the energy state within the brain. They're very responsive to things like ATP and ADP with their receptors such as P2RY12, P2X7R, I believe it is. And so they're these really dynamic cells with a number of different roles within the steady state CNS. To perform these roles, though, microglia are continuously surveilling the central nervous system. And this, I guess you could probably consider this a seminal paper in microglial biology. It's a few years old now, but what they find is that just within the homeostatic CNS, microglia are continuously extending and retracting their processes to monitor what's going on within the brain and spinal cord parenchyma. And so in this video here, you'll see in green is an example of the extension of microglial processes. In red, it's a retraction of microglial processes. So I'll get you to focus in this area here. You'll see a nice example of extension as well as down here. If it plays, oops, too far. Okay. There you go. So right in this area here, you can see nice green extensions as well as down here. So I hope you can appreciate that microglia are very dynamic. They're always moving their processes to sense the environment around them, which allows them to respond to any insult almost as soon as it occurs. And one last thing I want to touch on is microglial heterogeneity. This refers to the transcriptional phenotypes and transcriptional states that microglia can adopt throughout life. And what's worth noting is that these transcriptional phenotypes likely reflect distinct functions of those individual microglia. And as you can see here, heterogeneity is highest in embryonic and postnatal development, but once microglia reach the adulthood stage, they're largely homogeneous, and this is where they are doing kind of that, just surveying the brain parenchyma and just trying to identify any threats as they occur. And then with age, heterogeneity goes back up. There's some abnormalities that occur within microglia due to the aged environment, which we might see or we will get into a little bit later on. So it's worth noting that this heterogeneity isn't specific to microglia. Astrocytes also display quite robust heterogeneity throughout life, but we won't have too much time to touch on that. So just keep that in mind, is that these transcriptional state changes throughout life likely reflect distinct functions of these individual subpopulations. So that's enough about microglia. They're these highly phagocytic cells, very important in establishing and maintaining the steady state CNS. And as we'll see in a little bit, they also respond to several insults and can actually be a bit detrimental when they do respond in certain ways. So the other cell type I said I'd talk about are astrocytes. And these can be considered the stars of the CNS in that they quite literally look like stars when you look at them under a microscope. Astro cells kind of speaks for itself, but these also play a number of physiological roles. One of the most important of these is maintaining the blood-brain barrier. They do this with their end feet here in green. They cover almost the entirety of the neurovascular unit. So all of these blood vessels are covered by the astrocytic end feet to form the glia limitans. And this is really important to prevent the entry of undesirable cell types into the CNS that could or would likely promote some sort of immune response that could be detrimental to the organism. Outside of that, they support neurons. Quite often, this takes the form of trophic and metabolic support. and is important for the actual formation of these synapses, the maturation and modulation of synapses throughout life. And lastly, they're constantly interacting with immune cells. Predominantly at steady state, this immune cell is microglia. There's all sorts of different communication styles that we'll see in a moment, but they are these highly communicative cells that work within the CNS. And we also have peripheral immune cells, but that's more of a kind of disease or injury context where that comes into play. And we'll see that in a little bit as well. So when we consider microglia and astrocytes and the CNS as a whole, I kind of like to think of it as an ecosystem where no immune cell in the CNS functions independently of its neighbors or its environment. Whether its neighbors be the same cell lineage, so microglia interaction, astrocyte interaction, that certainly occurs, but also in terms of intercellular lineage interaction, so microglia on astrocyte, for example. One of the predominant ways that this occurs is through cytokine and chemokine signaling. So just using the astrocyte microglia communication again as an example, you can have communication between the two cell types where microglia secrete one factor onto astrocytes, astrocytes secrete another. This is continuously ongoing, very important for reciprocal growth and such. You can have synchrony where both cell types respond to the same same factor, and then they communicate with each other. Or there can be this, like, say, for example, a signaling pathway where one factor signals onto microglia, microglia then release a factor that signals onto astrocytes, or vice versa, factor onto astrocytes, factor onto microglia. So cytokine and chemokine signaling, very important for neuroimmunology. Outside of that, though, there's also physical contact between cell types that's very important for maintaining the CNS. Now, if you'll remember the video I showed just a few slides back where microglia are continuously extending and retracting their processes, that's what allows these contacts to occur. So in that same paper, found evidence of microglia here in green contacting astrocytes here in red. Neuronal cell bodies are these dark spaces here, and you can see the microglia kind of wrapping around and contacting those. And the astrocyte sheath in red here around a blood vessel where astrocytes are forming that glial limitans. And just as an example of that, in this video down here, if you look, you'll see this process contact the cell body, this one as well. I hope you can appreciate again that microglia are constantly moving their processes, extending and contacting their environment. And this is how they really are able to sense some sort of abnormality very rapidly and then initiate an immune response. And lastly, but not least, are exosomes and extracellular vesicles. I'm certainly no expert in this, but essentially the long story short of this is that astrocytes and microglia can both package cellular cargo into these small vesicles that they can then secrete into the environment, which can then act on their target cells throughout the CNS, whether it be neurons, oligos, microglia, or astrocytes. So again, a few different modes of communication, certainly not an exhaustive list, but these are some of the methods by which these CNS immune cells are able to interact with each other and maintain the CNS. But as I alluded to earlier, there's not just the central nervous system immune cells at play. There's also evidence for peripheral immunology interfacing with the central nervous system. The main ones among these are T cells. These have been found quite, well, not a lot, but they are present within, say, the subarachnoid space and within the cerebrospinal fluid. In a healthy young adult, there's not really any evidence of these being in the brain parenchyma, which is good. Once they get in there, there's generally something very wrong. But the roles of these are still somewhat unclear. Without T cells, though, there are neurocognitive deficits in mouse models and neurogenesis deficits as well. So they obviously have some sort of role, but it's still, to my knowledge anyways, not entirely clear what those roles are in the steady state CNS. So to this point, we've looked at microglia, or a few different things, but just to review what we've touched on, we've looked at microglia and astrocytes and how they are essential in the steady state CNS, both in its establishment and its maintenance. We've looked at how, very briefly, how cellular interactions can facilitate the maintenance of the CNS. And we've looked at very briefly how peripheral immune cells have somewhat unclear roles in regulating the steady state CNS. So this is all well and good. We have a healthy central nervous system, but things go wrong. Why do they go wrong? We don't really know. And because if we did, a lot of us would be out of a job. So there is that to consider. But what has been addressed much more, or what can be addressed much more easily, and what's working to answer this question is what goes wrong. And as I alluded to earlier, the biggest thing is that these immune responses don't resolve, which leads to a chronically activated response. This leads to quite robust damage within the CNS, quite robust inflammation. And that's not really the ideal scenario. In the case of MS, this leads to demyelination, or the damage of myelin on axons. Once that myelin is gone, it's no longer there to protect the axons, so the axons themselves can become damaged, which obviously is not an ideal scenario, considering that the repair and regeneration of neurons and axons is quite difficult. A number of different things also cause the damage: cytokines, glutamatic cytotoxicity, reactive oxygen species. All these things can contribute to damage within the CNS when these immune responses do not resolve. And so I'd like to, we've gone over a lot of the kind of fundamentals of I shouldn't even say a lot. We've gone over some of the fundamentals of neuroimmunology, because there is a lot more I'd like to touch on, but we don't have a lot of time. So I'd like to spend the last 5 to 10 minutes here going over some of what's new in neuroimmunology and what I think is exciting. Personally, I get very excited by studies that identify new cell-cell or cellular interactions, especially in steady state, but also in disease. And one of the big pushes here has been to understand how T cells interface with the central nervous system immune cells in disease conditions. So in a lot of work by Fran Quintana's lab, they've done a great job of identifying how T cells and astrocytes can shape each other's responses in a disease condition. So there is bidirectional regulation where, for example, you can get anti-inflammatory interactions where astrocytes can contact T cells and actually suppress them. But T cells can also signal onto astrocytes and suppress the astrocytic response in certain disease conditions. Or you can see T cell or astrocytes inducing T cell apoptosis, which could have benefits in certain scenarios. But on the flip side of that, you can also get an elevation of inflammatory responses, both with Th17 cells secreting, say, GM-CSF, which then promotes a reactive astrocyte response, or astrocytes signaling onto T cells to generate their own pro-inflammatory activities. So again, we're seeing increasing evidence in disease of these astrocyte T cell interactions. But my favorite cells, as I mentioned, are microglia, and there's been an increasing body of literature looking at how microglia and T cells interface within the CNS. Now, this paper is from Micah Simmons' lab, and it's pretty interesting. They show that, well, I guess I'll just show you the data, but they go on to show that within the aging white matter of the central nervous system, so this isn't a disease, it's just simply a 2-year-old mouse, so quite old for a mice, there is the presence of these CD8 positive T cells that emerge within the central nervous system. Let me grab a laser pointer here. So in green here, you can see the CD8 positive T cells that are now present within the CNS. But what goes alongside that is that there are these microglia stained here with IBA1 that are positive for STAT1, which is downstream of the interferon gamma signaling pathway, suggesting that these microglia are interferon responsive, which isn't something you normally see in a steady state CNS. And what was interesting here is that these interferon responsive microglia are very closely opposed to the CD8 positive T cells. So they're very close together. And you can see that the majority of them, around 60% of these interferon responsive microglia, are quite close to a CD8 positive T cell. And very few are just close to any other random cell within the aged central nervous system. And just as a further proof of concept, if you have CD8 knockout, so these T cells no longer develop, you don't get those same interferon responsive microglia in the knockout. So again, just suggesting that in a non-disease CNS, just as in an aging environment, T cells can influence microglial biology. On the flip side of that, though, a second paper that came out in late 2022 looked at microglia T cell interactions in EAE, experimental autoimmune encephalomyelitis, common model of multiple sclerosis. And what they find is that rather than, in this case, the T cells instructing the microglia, it appears that the microglia can actually instruct the T cell compartment. Just as a proof of that here, they did an image stream analysis. So this is flow cytometry, and they're taking pictures of these cell, I guess, aggregates, you could call them, as they pass through the flow cytometer. And what's really interesting is you can see here, there's clumps of 2 to 3 cells in all of these. And I'll draw your attention to some microglia markers like CD11B, TMAM119, ICAM1, MHC2, and then some T cell markers in CD4 and T cell receptor beta. And you can see they are coming through together, forming these aggregates, suggesting that there is some sort of physical interaction occurring between microglia and T cells in this model of EAE. And just on a similar note to the last paper where we were looking at interferon signaling in microglia, this particular interaction also appears to be dependent on interferon gamma signaling. So in these mice that are or have the interferon gamma receptor conditionally knocked out in microglia, we see a reduction in these CD4 positive T cells. So these regulatory T cells, they are not as prevalent when microglia are unable to respond to interferon gamma. So it seems that well, and I guess they go on to show that mice deficient in MHC class 2, which is important for showing antigen to the T cells, they also do not develop this regulatory T cell population quite as much. So again, just another example where microglia can interface with T cells in a disease condition, which I think is pretty interesting and neat. So we're rapidly running out of time for this morning, or for my part of the talk, so I'll just go through main takeaways. Again, I'd like to remind you that neuroimmunology, in case you haven't realized, is a lot more complex than what we touched on here, this little, I guess, tip of the iceberg, you could call it. But I hope you can appreciate that microglia astrocyte crosstalk within the CNS is fundamental in both physiology and disease. I didn't get into it too much, but this kind of interaction between these two cell types is a very prevalent response in almost any neurodegenerative or neurocognitive condition. But again, they do have very critical roles in maintaining the steady state CNS. And lastly, peripheral immune cells can both influence and be influenced by CNS immune cells, which I think will have several implications for neuroimmunology research down the line.